start immediately?
Yes. So
Okay.
I started. So the attendees are going to start coming in.
Mm-hmm.
Yes. So now we've started the webinar. I'm going to keep quiet
Okay.
and we wait for more people. And I'm making you co-host so that you also be able to see attendees as they come in.
Okay.
Okay. Thank you so much. You can
Thank
take
you,
over
Dada.
from here, Neza. As soon as you're ready, you can give it a go. Thank you.
Yeah, it's 10:58 here, so
Yeah,
maybe
we
I will
still
start.
have quite some time.
Yeah.
Definitely. Yeah. Okay. Thank you.
Mm-hmm. So I think we can start. It's 11 sharp, 4 sharp for Morocco. So welcome everybody and thanks for joining us today. Please join me to welcome our panelist, our speaker, Dr. Zakaria Ouhaz, who is an assistant professor of physiology at Institut Supérieur des Professions Infirmières et Techniques de la Santé. For English speaker, it's Health Professions Institute, belonging to located in Marrakech, Morocco. Dr. Ouhaz holds a PhD in neuroscience from Qadi Ayyad University, and he completed a postdoctoral training at the University of Oxford. He is a Royal Society Newton International Fellow investigating thalamocortical interaction in cognitive processes, including learning, memory, decision making. His research uses behavioral testing, electrophysiology, brain imaging, and neuroanatomical techniques to examine how the mediodorsal thalamus contribute to cognitive function, including studies on cognitive effects following thalamic damage. Dr. Ouhaz, thank you for sharing with us today your work about functional dissection of the mediodorsal thalamus in pain processing. The screen is yours.
Thanks very much, my dear friend Nizar, for the kind invitation. So I would like also to thank the organizers of Sona webinar series for giving me the opportunity to present to you today a project that we are currently working on. So most of the data that I will be showing today are not yet published. So it's like you are the first one to be exposed to this kind of to these data. So my talk for today, during my talk, I would try to convince you that the mediodorsal thalamus is implicated in pain processing, and that the mediodorsal thalamus, as it is composed by three subregions, each of these subregions is implicated in pain processing in a different way. So as a start, I would like to give an overview of the neuroanatomy of the thalamus. So the thalamus is this, basically it's this part. It's a gray, a large mass of gray matter, which has a size and shape that resembles the small hen's egg. And the thalamus is not a homogeneous structure. We can identify different thalamic groups. So like we have the anterior thalamus, the lateral, but it's located in the ventral part. So it's the ventral lateral part, the lateral part, and the medial part with the mediodorsal path and the most posterior region of the thalamus, which is called the pulvinar. We should highlight that all the nuclei of the thalamus except the reticular nucleus project to the ipsilateral cerebral cortex. So we have few contralateral projections from the thalamus. So mainly like 90% of the thalamic projections to the cortex are ipsilateral. And the whole of the cortex receives inputs from the thalamus. And all thalamic nuclei receive corticofugal fibers in a reciprocal fashion. So we can classify the functions of the thalamus either to the thalamus is implicated in motor behavior and also in emotion and cognition. And we know that the thalamus analyzes all the sensory and cognitive and also emotions that needs to be analyzed and treated by the cortex. When it comes to thalamic lesions, patients that have thalamic patients, they have Hall marker phenotypes. So they have a loss of sensation in the contralateral side of the face. So what is called the thalamic pain. So the thalamus is known to be involved in pain processing. The thalamic syndrome, for example, which is characterized by abnormal voluntary movements, what we call the chorea, with hemisensory disturbance. In order actually to understand the physiopathology of the disorders implicating or involving the thalamus, we need to understand the thalamocortical connectivity. So there is the work done by these two guys, Gallery and Sherman, that showed that thalamic connectivity is not as we've been taught in textbooks. We have the classified thalamic inputs into drivers and modulators. They're not just, we don't have only afferents that will only convey sensory inputs to the cortex, but we also have thalamic projections that will be implicated in the modulation of the cortical computations. So, for example, we have a specific relay neuron, and this specific relay neuron will send projections to the primary cortex and especially to layer 4. So, for example, if we have the LGN, so the LGN will send the projections to layer 4 pyramidal cells. What we need to take on consideration is that these afferents have also collaterals that will innervate the layer 6. So mainly specific relay neurons, they will send projections to layer 4, and the most important projections will arrive to layer 4, but we have few contralaterals that will also give inputs to layer 6. Then we have non-specific relay neurons that will only send projections, non-specific projections to upper layers, mainly layer 1, 2, and 3. And the corticothalamic projections originate from layer 6. So we have the thalamus that will send projections to layer 4 and layer 6, and the output of the cortex will originate from layer 6. So the question is, how does the information get down to the cortical layer 6? So basically we have neurons originating from layer 5 and also in neurons from the upper layers that will send projections will receive projections from layer 4 to layer 5, and then from layer 5 to layer 6. And layer 5 here is very important because it will also give birth to corticothalamic projections. And these corticothalamic projections will innervate another set of thalamic nuclei, what we call the higher order nucleus of the thalamus. Okay, so it's like if we will have higher inputs to the cortex arriving to layer 5 to layer 4, and then from layer 4 we will have a modulation of these afferents via the implication of the higher order thalamic nuclei. So it's not something that is vertical, but we have a lot of loops and a lot of interactions between the different layers of the cortex and the thalamus. So the higher thalamic nuclei will receive, will process higher level information, because these information have been previously analyzed and processed in layer 4 and layer 5. Okay, so the thalamic nuclei are classified according to this pattern of connectivity, either to driver cells. So the driver cells are the one that will send projections to layer 4, or modulator cells or modulator nuclei that will receive the processed informations from layer 5 toward the thalamus, and then from the thalamus again to layer 5 of the associative cortex, which could be, for example, the prefrontal cortex. So, as an illustration of this driver and modulator, if we see the picture by the New York Times about the Tour de France, you can see that we have clear image of the cyclists. This clear image is due to the intervention of the modulator modulator thalamus of the high order thalamic nuclei. The image that is not clear, it's only the driver. So the driver will send the image to the cortex will process it, the layer 4, and then from layer 4 to layer 5, then from layer 5 to the higher order thalamic nuclei. So the clear image is a combination of the effect of the activation of the driver plus the modulator. The unclear image is only the result of the driver activity, because there is no modulator. Okay, actually it's a process that is also implicated in attention and also in high order cognitive functions. So the mediodorsal thalamus is considered to be a higher order thalamic nuclei. It is implicated in cognition and also in emotion, and damage to the medial temporal lobes of the cortex results in deficits. For example, damage to the medial diencephalon would have like the infarct patients, or by stroke, we can have the Wernicke-Korsakoff syndrome. And the changes in thalamocortical connectivity was linked to neuropsychiatric disease and neurodegenerative disorders like schizophrenia, obsessive compulsive disorders, Alzheimer's disease, behavior variant frontotemporal dementias, Parkinson's disease, alcoholism, and major depression. So the mediodorsal thalamus is a hallmarker of cognitive disruptions related or symptoms related to many neuropsychiatric disorders or neurological disorders. So as I said, the mediodorsal thalamus is a higher order thalamic nucleus. It is critically involved in cognition and emotion. And the mediodorsal thalamus is composed of 3 subregions. We have the lateral part here, the MDL, the MDC, the central part, and the mediodorsal thalamus. And we have a cytoarchitecture difference between these three subregions. It's not only anatomically distinct, but it's also in terms of the cellular composition, it's not the same. So the medial part is composed of densely packed multiple neurons. The central part, it's heterogeneous, and the density is intermediate. However, the lateral part is small, sparsely distributed neuron. It's composed by small and sparsely distributed neurons. So this anatomical heterogeneity, does it mean that we have a functional dissociation between these subregions? As I said, the mediodorsal thalamus was defined, historically was defined as the part of the thalamus that sends projections to the prefrontal cortex. So the prefrontal cortex receive reciprocal and extensive projections, glutamatergic projections from the mediodorsal thalamus. And we do have a kind of parallelism between the cytoarchitecture of the mediodorsal thalamus and the prefrontal cortex. We know that the medial part sends projections to deep layers of the prelimbic cortex, the central part to the superficial and deep layers of the prelimbic cortex, and the lateral part sends projection mainly to the anterior cingulate cortex, mainly in layer 1 and 5. And the important thing to highlight here is that we will find the cytoarchitecture of the mediodorsal thalamus in the prefrontal cortex. So we have a selective functional selectivity. So the prefrontal cortex and the ACC are not involved in the same way in terms of pain processing. It has been shown that the ACC is more implicated in affective pain, and the prelimbic is more implicated or involved in cognition and action set. So our hypothesis is that if we silence these subregions of the mediodorsal thalamus, we would have different responses to pain. If we disrupt for example, the MDL, we will also disrupt the integrity and the function of anterior cingulate cortex, and we will have a phenotype that is different than, for example, inhibiting the medial and the central part, that will give us a phenotype that resembles more to the dysfunction of the prelimbic cortex. So that was our hypothesis. It's that we know that the prefrontal cortex is implicated in pain processing, but our hypothesis is that we do have a kind of processing of painful or of nociception in the mediodorsal thalamus before these informations arrive to the prefrontal cortex. We don't think that the prefrontal cortex is doing all the computations, especially because the mediodorsal thalamus, as I explained before, is a modulator. So the modulator will give like a sort of preparatory set to the prefrontal cortex to be ready and to interpret the best way the stimulus that it receives. So we suspected the functional heterogeneity. It has been previously shown many years ago that the mediodorsal thalamus is implicated in pain. So in animal models of chronic neuropathic pain, mediodorsal thalamus as a whole nucleus had presented with different patterns of activation that presented like a signature, electrophysiological signature, that the mediodorsal thalamus is involved in processing nociception. So now the question is, do the the subdivisions play distinct roles in pain processing. To assess this question, we did two things. The first thing, it was like a preliminary study, was to target these subdivisions, the lateral part, the medial, and the central part, by excitotoxic lesions. It was via the injection of NMDA to these subregions. It's a kind of tricky surgery to do because the thalamus, the mediodorsal thalamus, is a small structure, and we needed to have to get our stereotaxic coordinates that will allow us to specifically target these subregions. So what we did is we silenced the lateral part, but for the medial and the central part we couldn't, because the two structures are too close, and they overlap in some in the anteroposterior axis. Then after the lesions, we performed, and after the recovery of our animals, we performed many behavioral tests, mainly the Van Frey for mechanical pain, the hot cold plate for hot and cold pain, and the place escape avoidance paradigm to assess the emotional component of pain processing. The two first sets of the behavioral testing, which means the Van Frey and the hot cold plate, they were performed to assess the discrimination that our mediodorsal thalamus can discriminate, can identify a painful stimulus. And the place escape avoidance paradigm was used to assess the emotional component of pain processing. Then after completion of the task, we perfused our animals, and we processed our brain tissue to verify the lesion or the injection sites. So concerning the Van Frey, the mechanical pain, we see that male and female rats, they present a decrease in their withdrawal threshold, showing that they have developed sensitivity to mechanical stimulus. The same thing with the hot and the cold plate. So the lesioned animals, they tend always to show a decrease in the withdrawal latency, which means that the medial part and also the lateral part are both implicated in pain processing, in identifying the painful stimulus. And the thing that I want also to highlight is that male and female rats are not processing the noxious stimulus the same way. Female rats are more sensitive to mechanical and thermal nociceptions, and when we perform the lesion, this dimorphism will vanished. The male and female rats after MD lesions, either to the mediocentral part or to the lateral part, will respond the same way to the noxious stimulus. Concerning the place escape avoidance, here we have the sham, the MDMC, and the MDL. And as you see, because here, please, I would like to that you make a little bit more attention, because it's kind of tricky to understand. So when we lesion the mediodorsal, the mediocentral part of the MDL, it means that this part is missing, and the other part, which is MDL, is functioning. When we lesion the mediodorsal lateral part, the mediodorsal lateral part is missing, but the MDMC is well functioning. Okay, so for example, if we take the male rat, what we have is that when we perform MDMC lesion, we have an increase in the time spent on the light side, which means that our rats have developed an aversion because usually they will tend to prefer the dark component compared to the light or to the bright compartment. With the lesion and also the application of the painful stimulus, these rats after MDMC lesions, they will tend to spend more time in the bright compartment compared to the dark compartment, okay? Which means that MDMC is not existing, it's lesioned, but MDL is there, which means that the phenotype we're seeing here is 100% the result of the activation of MDL, okay? When it comes to the MDL lesioned group, we lesion the MDL, we have a similar pattern as for the sham group that didn't receive any lesion, and the same pattern was replicated in female rats, which means that only the medial lateral part of the lateral part of the mediodorsal thalamus is implicated in the emotional component of pain, because here the aversion we're seeing is only the result of the activation of MDL because MDMC is missing. Here MDL is missing, but MDMC is there, okay? So the first three conclusions are that despite hyperalgesia, the MDL group showed reduced aversion to pain in the dark box. MDL subregion could be driving the aversion, and the MDMC is not important for aversive behavior, okay? So as a first sum up, we have functional dissociation of MD subdivisions. The three subdivisions, or at least the two, because we combine the lesion of the medial and the central part, they are not doing the same tasks in terms of pain processing. However, MDMC and MDL are both implicated in sensory discriminative processing of pain, because MDMC plays a prominent role in the perception of pain intensity. MDL is the only subdivision that is implicated or involved in processing the affective aspects of pain. And it's also implicated or involved in the sensory discriminative process of pain. Okay, now we have demonstrated that the mediodorsal thalamus is implicated in pain, and that the different subregions are also implicated in different ways. Each part is responsible of a kind of processing. The MDMC is implicated in the discriminative processing. MDL is more implicated in emotions. Now, we were interested in studying the connectivity, how the connectivity between the mediodorsal thalamus and these subregions with their projecting areas in the prefrontal cortex are driving affective pain. And the first study, what we've done, we were interested in evaluating the connectivity between the mediodorsal lateral thalamus and the cingulate cortex in terms of affective pain. So what we've done, we used optogenetics. So we injected two viruses. One, the ARC-T, to inhibit thalamic projections toward the ACC, and we used channel rhodopsin 2 to activate. So we did both manipulations, inhibiting these projections and activating these projections. And we injected in the thalamus, but what we did is that this virus was combined also with tracers. So we evaluated the expression within the ACC to be sure that these afferents are mediodorsal lateral part to ACC, and we performed the optogenetics in ACC. So we inject in the lateral part, and we put the laser to activate or to inhibit in ACC, and we're sure that we are involving only this pathway. And as you see here, so these are the lesion sites within the MDL, and you see that it's very specific to the lateral part. And you can see also the expression of the virus in the ACC, meaning that these viruses were arrived to ACC, and they were expressed. So okay. So we performed again the three tasks. So the Van Frey, hot plate, and cold plate to determine the involvement of the mediolateral part in the discriminative processing of pain stimuli. And what we have, we assessed the behavior of our animals before and with the laser on and after recovery. And as you see, that when we inject in the lateral part and we activate the ipsilateral ACC, we have a decrease in the threshold of Poe withdrawal, meaning that we have an increase in the sensitivity, in pain sensitivity. The same result was reported in the when we activate the contralateral part. Even if I say that the thalamic projections are mainly ipsilateral, but this effect of the contralateral, we suspect that it is due to the corticocortical thalamic interaction. Corticocortical interaction, not involving the thalamus at all. The same thing in female rats. So in the females, the activation, the inhibition of MDL projections to ACC will decrease the threshold of Poe withdrawal. The same thing with in the hot plate and also in the cold plate, meaning that when we are inactivating these projections, we have similar pattern. We have a decrease of latencies and of the withdrawal threshold, attesting an increase in pain sensitivity, meaning that selective inhibition of these projections decreased pain thresholds in the Van Frey test of the ipsilateral and also in the contralateral Poe in rats. Then we did the same thing with the MDMC subregion. We inhibit the MDMC projections to ACC, and we have we reproduced the same effect. So both projections, MDL and also MDMC projections, are implicated in the discriminative processing of pain in interaction with ACC. So ACC is implicated in the discrimination, and both MDL and MDMC projections are implicated in the discriminative component of pain processing. So when we inhibited with ARCT the projections between MDMC and ACC, we have an increase of the time spent in the light side, which means that we have an aversion. So the rat tend to stay in the bright side more than staying in the dark compartment which is associated with the painful stimulus. When we inhibit MDL projections, our rat perform as sham, as without any light on. Okay, which means that again, when we inhibited MDMC, the MDL was still intact, was still functional, and when we inhibit the MDL projections, MDMC was activated. Okay, so only we confirmed again that only the MDL ACC projections disrupt the emotional response to pain. So persistence in the dark compartment during the task of place escape avoidance paradigm, even if we have ongoing nociceptive stimulation and hyperalgesia. Okay, and the same thing was observed both in male and female rats. Then we wanted to test the activation. What about the activation of these afferents if we will have the opposite effect that we had when we silenced or when we inhibited these projections? So with the Van Frey, when we activate these projections, we have an increase in the threshold withdrawal, which is the opposite. So we have a sort of desensibilization of our rats, which is the opposite of what we had when we inhibited, which confirmed the results, the previous results, and that these afferents are involved in pain processing. The same thing with the hot and cold plate task. So again, the results of activation confirm the results of inhibition. So activating MDL ACC projections elevates mechanical and thermal pain thresholds bilaterally in both sexes, indicating a robust anti-nociceptive effect. We did the same thing with MDMC. And with the MDMC, what happened is that when we activate MDMC ACC projections, we have the same results as if we inhibit those projections. We have an explanation why, because we think that MDMC projections received a reverberation, like negative feedback from the cortex that will inhibit the thalamic drive, and we will find the same thing as if we are inhibiting. So basically, with the MDMC ACC projections, we have the same results in the Van Frey hot plate and cold plate task as if we inhibited these projections. So MDMC ACC activation further decreased mechanical and thermal thresholds in both sexes, mirroring the effects seen with optogenetic inhibition. When it comes to the emotional activation, we have the same thing. So when we activate mainly the MDMC projections, we will get the same effect as if we inhibit MDMC. So showing that again, MDL projections are more implicated in the emotional component of pain processing. Okay, and when we activate MDL, we don't have any. Our animals will have similar behavior as with before activating. So activating MDL ACC projection disrupted the emotional response to pain. That's the conclusion of these experiments. So the first take-home message is that we have a functional specialization within the mediodorsal thalamus. MDMC and MDL are functionally different in pain processing. The projections from MDMC to ACC pathway drives the sensory discriminative dimensions, controlling stimulus intensity and localization. And MDL-ACC pathway controls the affective motivational dimension, shaping aversive responses to pain. So convergent evidence is that MDL lesions and optogenetic silencing of MDL projections to ACC increase hyperalgesia and pain aversion, and that we have a circuit causality. So optogenetic activation of MDL ACC restores normal affective responses, highlighting this pathway as a therapeutic entry point and a marker for chronic pain. As I've shown here, when we activate the MDL, we have a behavior that is similar, or pain sensitivity that is similar to when we don't have any activation. So we have the MDL, if we, as a therapeutic intervention, if we succeed to activate the lateral part, we will be able to have a normal behavior of our rats or our patients suffering from chronic pain. Then we've been interested in studying the effects of the modulation of the mediodorsal thalamus projections to the prelimbic cortex, or to the prelimbic sub-region of the prefrontal cortex. We did the same thing. We targeted the MDMC aggregate and the lateral part of the mediodorsal thalamus with both viruses. We activated and inhibited these projections, and we assessed our animals in the same battery of behavioral tasks, and we injected in the MDMC or the MDL, and we activated the prelimbic cortex. So the light was shed on the prelimbic cortex, either to activate or inhibit this connectivity. So concerning the data of the Van Frey and hot and cold plate tasks, we have shown that when we inhibit MDMC projections to the prelimbic cortex, we have a decrease of the threshold of PAO withdrawal, meaning that we have an increase of pain sensitivity, both in female and male rats. The same thing when we inhibit MDL projections to the prelimbic cortex. We always have a decrease in these in these thresholds, meaning that these projections, the MDL, both MDL and MDMC projections to the prelimbic cortex, are implicated in the discriminative component of pain processing. Okay, the only thing that I want to highlight is that the MDL prelimbic cortex afferents are more implicated in cold pain compared to male rats. So here we don't have any significant difference, but with female rats we have this kind of difference. The males are more sensitive to cold when we activate the mediolateral part projections to the prelimbic cortex compared to their corresponding female experimental group. So both MDMC, again, and the conclusion is both MDMC and MDL are implicated in sensory discriminative processing of pain. Then what we have done is that we activated. So after we've inhibited, we activated, and we've seen that MDMC projections to the prelimbic, even if we activate, we have the same phenotype as if we inhibit. And again, this is due to these projections from layer 3, GABAergic projections that will inhibit the MDMC, and then we will have, when we activate MDMC, we will activate the cortex, and then the cortex will inhibit MDMC, and we will find the same phenotype as if we are inhibiting this pathway. Concerning the emotional processing, when we inject GFP and we try to activate the prefrontal cortex, we won't have any effect. So we are sure that it's a control experiment to be sure that our what we are seeing is due to the viral expression. Okay, so when we inhibit with ARCT, when we inhibit MDMC and MDL projections to the prelimbic system, cortex, we have a similar pattern. So meaning that these afferents are not implicated in are implicated in pain processing the same way. However, when we activate these projections with MDMC, we have always the same phenotype. We have an increase of aversion. When we activate MDL, we have a decrease, and we have a pattern that is similar to a sham group, a group that is not activated, which means that MDL projections to pre-limbic cortex could be implicated also in the emotional component of pain processing. So both in female and also in male rats, we have basically the same results. We don't have any sex differences. So the take-home message of my presentation is that we do have functional specialization of the mediodorsal thalamus. So MDMC and MDL are functionally different in pain processing. MDMC toward prelimbic, this pathway drives the sensory discriminative dimension, controlling stimulus intensity and localization. The projections from MDL to prelimbic cortex drives the sensory discriminative dimension, controlling stimulus intensity and also localization. They have the same effect, but only MDL, ACC, and MDL prelimbic projections are implicated in emotional processing. So, contrary to what we were expecting, that only MDL, ACC are implicated in pain processing, MDL prelimbic cortex afferents also are implicated in this type of processing, which means that the MDL is the modulator of emotional processing rather than the medial or the central part. It doesn't matter to what subregion of the prefrontal cortex these afferents arrive. The MDL is the hub. We have like a sort of the first filter within the thalamus, which is this dissociation between MDMC and MDL. So MDL projections toward ACC or prelimbic cortex are implicated in emotional processing. So this my presentation is finished. But before, I would like to thank Hanan Ibn Dawdi. She is the PhD student in our lab that is conducting these experiments. So this work is a part of her PhD project, and she did an amazing job. And I would like also to thank Prof. Mark Laundry. So all the optogenetic manipulations were performed at his lab at the University of Bordeaux. And as always, I would like to thank Professor Mohammed Benis, who is my mentor and also the director of our laboratory. So and I want also to thank our funding bodies. This project was financed by a project by the European Commission, Psychomed, and also with Marie Curie actions. I'm personally supported by the Royal Society, and I would like to extend also my thanks to the Society of Neuroscientists of Africa for giving me this opportunity to share this work, this amazing work that Hanan did with you. And thank you for your attention, and I'm ready for the question. Thank you.
Thank you so much, Dr. Ohaz, for this very interesting webinar talk. So I open the floor for the participant to ask questions, either by writing them on the Q&A session or raising their hand, and then we will activate their mic. I see now that Dr. Lile Kulu joined us. So are there any questions? So we have one question. Yes, go ahead, Al-Hafid.
Thank you very much, Professor Nizha, and also our dear colleague Zakaria. I have no question. I just want to say that that was very helpful, and thank you very much.
Thanks very
Thank
much,
you,
my
Al-Hafid.
dear friend.
We have another thank you from Bilal Mansouri, who says, many thanks, Dr. Zakaria.
Thank you, Bilal. You're welcome.
So I have a question. I have so many, actually. Yeah, I was like confused when you are talking about when MDMC is activated. So it means that it's not involved. So it was like confusing. But the question I thought about at that moment, is there any connection between these two regions? Is there any action, inhibition or activation between the medial dorsal, centromedial and the lateral one?
Actually, no, there are no evidence that the subregions are interacting, because we would expect that we could have interneurons between the different subregions, but in the thalamus, the only GABAergic part is the reticular thalamic, which is away from the mediodorsal part. And usually the mediodorsal thalamus is considered to be like one structure, one solid structure. There is no interaction. The subregions are doing the same thing. But now we show that no, they're projecting to different areas. So even though we've seen before, like at the end of the presentation, that both MDL sends projection to both to the ACC and also to the perilimbic cortex, and the same thing with the perilimbic cortex.
MC.
Yeah. But the prominent projections from the lateral part are toward the ACC, not toward the prelimbic. And the MDMC, the major parts and projections are to the prelimbic cortex. So we have this kind of specificity in terms of projections, in terms of axons projections, and there are no evidence that there are any interactions between the two structures. When we lesion the lateral part, we're sure that the lateral part is not there.
Yeah, I understand. I don't know. It's more confusing since I know that the prelimbic cortex is more involved in aversion response, right? So, and when you say the MDMC is like less involved in aversion or response or discrimination, I'm confused there.
Because it depends, because actually when we have the connectivity, the cross connectivity is between ACC and prelimbic cortex.
Mm-hmm.
So what we and also it could be like a sort of task specific, because when you are we're usually talking about aversion, it's when we're performing learning
Yeah. Mm.
protocols
Yeah.
that
Yeah.
are
Exactly.
usually they take longer time than with the 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 task we performed. When we for example we inactivate the prelimbic cortex, because it's a specific it's specific targeting. So we inject the virus in for example, the medial part,
Mm-hmm.
and we put the laser in the prelimbic cortex, and we activate. So when we're activating the whole
The
circuit,
pathway. Yeah.
the pathway. So this particular pathway is not implicated in
Yeah.
aversion, and the prelimbic cortex receives other projections from the hippocampus, from the amygdala, that could be the results of or the key structure implicated in the aversion you're talking about.
Okay. We have a question from Bilal. Your
Hello?
mic is activated.
Hello, can you hear me?
Yes.
Yes.
Yeah, first of all, I would like to thank you, Zakaria, for this nice talk. Just wondering, how can you explain the difference between males and females in terms of perceiving anxious stimuli? Thank you.
Thank you for your question. It's a tricky one. We don't have an explanation, to be honest. What we've shown here is that even in sham groups, female are more sensitive to noxious stimulus compared to male. But when we lesion the mediodorsal thalamus, like I'm talking about the lesion studies, this effect will just vanish. The male and female will, with MD lesions, will respond exactly the same way. They will have basically the same thresholds. And also when we activate or inactivate the projections, we don't see much of this gender difference. Yeah, I don't have an explanation, to be
Yeah,
honest.
actually, I see it's hard to explain in terms of sex difference, and that's why I thought maybe you have an explanation. Also, I don't know. Yeah. Thank you.
Cheers.
Yeah, it's very complicated
To explain.
to explain sex differences, because whenever you do an experiment using both sexes, you end up having different results. We have this tendency to blame the sexual hormones,
Hormones.
but we don't know the actual mechanisms of these differences. Is there any other question from the audience? Okay, another question for you, Zakaria. Do you have
Yeah,
any idea of the neuronal subpopulations in these two regions?
they're all glutamatergic.
All?
All, yeah.
Okay.
Yeah,
So
they're,
all
yeah,
stimulatory.
yes. They're activating.
Yeah. And another question, like for these two regions play the role of the modulator structure that you talked about in like when introducing, right?
Yeah.
So to perform this modulatory action, this region gets information, either sensory or cognitive information from other structures to be able to control or to give like the right direction to the ACC and prelimbic cortex? And if it's the case, which are these regions?
Well, actually, the projected areas to both structures are, we have projections from the hypothalamus, from the basal ganglia, we have reciprocal projections from the amygdala, and also from the hippocampus. So and also from the reticular thalamus. So the GABAergic control from the TRN. We have basically the mediodorsal thalamus will receive a whole bunch of information coming from different subcortical regions, and also from the cortex, because the thalamocorticothalamic projections are reciprocal. And as if you remember the scheme about the afferents to the modulator afferents coming from layer 5.
Yes.
So the thalamus will send back thalamic afferents to the same layer, to layer 5. That's why it's a modulator. So if we have another stimulation, layer 5 is ready.
Yeah.
And will perform higher computation compared to the previous exposure.
So I think we don't have any more questions. Bilal, you are still raising your hand. Do you have a question? Yeah.
Yes, I do.
Yeah, sorry.
Okay, thank you. Dr. Zakaria, sorry,
Oh,
I
you're
do have
welcome.
another question.
Yeah, you're welcome.
Okay, so the thalamus and both nuclei that you talked about, they are only involved in processing sensitive information, or they are involved also as a response to decrease pain threshold and stuff?
Sorry, I didn't get the question. They're both implicated in
only sensitive information, I mean from the thalamus to the cerebral cortex, or both directions, from the cortex also to the thalamus? And
actually, the mediodorsal thalamus is more considered in the ascending pathway, so from toward the cortex. But because then from the cortex will implicate other structures. For example, like if we take for another example, which is different, for example, anxiety. So we have all the sensory informations coming toward the mediodorsal thalamus, and then from the mediodorsal thalamus to the cortex, to the prefrontal cortex, and then from the prefrontal cortex, we would have the implication of the amygdala. We will have projections coming back to the mediodorsal thalamus, but it's just a kind of loop that will keep the prefrontal cortex ready to perform the adequate or adaptive responses. Okay? It's like if you would have mediodorsal thalamus to the prefrontal cortex to the thalamus, and then it's a kind of loop. It's like if you are updating the prefrontal cortex.
okay, perfect. Thank you.
Cheers.
I have a last question.
Yeah.
So in your model, it's thermal initiated pain.
Yeah.
Can you generate this like pathway? Is it general to all pain experiences, or is it specific to pain originated from thermal stimulus?
We did also the mechanical with the Van Frey.
Yeah.
So
Sorry.
it's to both.
Yeah, I'm sorry.
No, no worries.
Stupid question.
No, it's the presentation was full of that.
So any more questions? Okay, thank you so much, Dr. Ohaz, for your presentation. Thank you for all for Bilal, Hafid, and all the other attendees. And hopefully we can see you on the next webinar. Thank you again.
Thank you very much. Thank you very much for you, Nizar, for the kind invitation, and
Yeah.
also all the attendees for the interest they give to
Okay.
my presentation, and I hope to, yeah.
I'm sorry, I just
No
saw a
worries.
question on the question
Okay.
and answer.
Okay.
I'm sorry, Kimberly Williams just asked a question. So thank you for the interesting talk. Could you model your model be applied to investigate interoception?
Interoception is different because we have with interoception we do have two kind of ascending pathways. We have uh, s- uh, the spinothalamic pathway, and we have also the cranial nerves, mainly the 10th nerve, which is sensory and also motor nerve. Um, I'm not sure that the model will be applied in this kind of to this kind. Otherwise, we can perform we can cause gastric malaise for our rats. We can just inject lithium chloride. And then we need to perform electrophysiology to show that the mediodorsal thalamus is implicated. To be sure to have a signature that the mediodorsal thalamus is processing this kind of, it would be a different set of experiments.
I hope this answers your question, Ms. Kimberly Williams. Okay, thank you again, Zakaria. I'm sorry for cutting
No
cutting you
worries.
before.
Thank
Just
you
I
very much.
noticed this question at the last moment. So go ahead.
Yeah, thank you very much for Sona for giving me the opportunity to give this presentation, and hope to see you soon again with a different story, maybe different project. And thanks, Nizha, for the amazing job you're doing with Sona, and also the other jobs you're doing.
Thank you.
And
Thank
thanks
you so much.
to Bilal, Hafid, and all the other attendees for the interest, for staying with us, and for the interest they shown to my presentation, to the work we're conducting.
Thank you. Thanks, everybody. Bye.
Thanks. Thank you, everyone. Okay. See you.